Very nice to be here today. Very interesting crowd, although a lot of them left for lunch, I guess. So we're here with a few people left. I'm joined here with by my CDO, Kees Groen, and our Chief Business Development in the US, Kevin Elliott. My name is Marcel Jacobs. I'm the CEO of Matisse Pharmaceuticals, and I'm going to present to you the story of Matisse. I'm going to try to get the slides up, but it's a little bit tricky with the technology at the moment. We don't get it. That's really, there we go. Do you see that or not? No?
I don't see it. You just have to do a share.
Yeah, I'm doing that actually.
Press on the slide.
Yeah.
And then
Okay.
it says share and you're
Yeah.
up.
Okay. Excellent. Oh, not. I have it on. Sorry for the slow start. Basically, I can. Yeah, no, I will start. I mean, what you see actually, what I see on my screen and you don't see is a ticking clock on European time, because we are talking about sepsis. And when we talk about sepsis, every second counts. Matisse Pharmaceuticals is a Dutch biotech company focused on developing the first life-saving treatment against this very terrible disease. For those of you that don't know sepsis, the definition of sepsis is that there's a life-threatening organ dysfunction caused by a dysregulated host response to an infection. Now, I don't think there are lots of biotech investors in this room, so I may want to simplify that a little bit and make it easy for you to understand. What is sepsis, basically? Sepsis is the body's extreme reaction to an infection. So it's not actually the infection that makes you die at the end of the day, but it's the reaction to the infection. And that's also why for some people it's not so known. I will show you some numbers later on how many people are affected by that. So the common triggers of sepsis are bacterial infections, fungal and parasitic infections, viral infections, and trauma. This all can lead to sepsis. For about 11% of the patients, it's not exactly known why people develop sepsis. It's the leading cause of death worldwide. This a little bit depends on how you classify the different cancers, for example, heart disease, but in principle, it's the leading cause of death worldwide. And that is often very much surprising people. 49 million cases per year. 40% are children under the age of 5, which is also something that a lot of people don't realize. It's actually affecting the most vulnerable population. 11 million people die every year, and 1 out of 5 deaths worldwide are related to sepsis. That means that 1 person dies every 2.8 seconds. In this 25 minutes that I'm allowed to present here, thank you very much, Marty, we have 535 people dying. And by the time you go to bed tonight, I don't know what time exactly you go to bed, but it will probably be around 14,000 people who have died of sepsis today, from now on. Is it a developing world disease? Yes, in the developing world, the issue is even slightly bigger than in the developed world, but also in Europe and in the US, almost 1 million people die of sepsis every year. And this is the real issue. There is no effective treatment to treat sepsis. The average cost on top of that of treating sepsis patients are very high. So a sepsis patient that ends in the ICU costs on average 30,000 euro per patient, but that's only the small part of the cost when the patient is in the ICU. There's a lot of cost also afterwards. There was a recent study published by Buchmann et al. In the Journal of Critical Care Medicine that estimated the total healthcare cost in the US alone to be $62 billion annually. And these are the direct costs, these are not the consequential costs. So this is huge. So, now we are there. Okay. Now, what do we do at Matisse? We believe that we have a medical breakthrough with our program M6229. That doesn't mean 6229, you know, if you're in biotech, that's usually a code name for the amount of projects you have. We don't have 6,229 projects in development. It's actually the postal code of the University of Maastricht. And that's where this product was originally discovered, and we licensed in the technology from the university, and they're still very much also involved in further advising us to progress with this. What is M6229? M6229 is the first treatment that is focused on neutralizing extracellular cytotoxic histones. And thereby, we basically stop the septic reaction. I will go into detail a little bit more. For those of you who don't know what are histones, histones are positively charged proteins that are in the cell nucleus, and they have a function. Basically, they act as spools around which DNA winds, and they also play a role in gene codation. No problem with those histones unless there is a septic stimulus reaction. And that causes the activation of netosis, amongst others. It's not only netosis, but that's one of the main triggers. That means that those histones are released from the cell nucleus into the extracellular space. And the moment that happens, these histones become very cytotoxic, and they start damaging other cells, and you get a chain reaction of ever more increasing histones in the bloodstream, leading to tissue damage, organ failure, and multiple organ failure, and ultimately death. Here you see a little bit a different representation of those histones. You are not seeing that, I think. I see that. Let me see. Let's see if we can get it. I think now we're there. Yeah, you're a little bit slower than my computer. Here, so here you see a little bit what I just explained. The histones being released at the time of infection, and then more histones are released through various cell death types. So you get more and more histones in circulation, ultimately leading to organ failure. That's in fact where we start with our therapy right now. When a patient is admitted to the ICU, we are starting our M6-9 infusion. Is that the right time? We often get this question. Yes, for the current stage of development, it's the right time. But of course, you would like to start as quick as possible with this treatment, because the earlier you catch those histones, the better it is, because you have less underlying damage in the patient. Here you see a different representation of the histone release, and I also use this slide, if it comes up for you, to explain a little bit where we are and where other companies are or have failed, because there have been quite a lot of failed sepsis treatments in development. If you look at the upper part of the picture, that's basically the trigger. So you have the infection. A lot of companies are focusing on treating the infection, but that's not always effective for two reasons. The main reason actually being that the septic reaction can already start at the time of the infection. So treating the infection itself doesn't stop the septic reaction. So a lot of treatments that are focused on curing the infection are not covering sepsis, even though they are often presented as sepsis developments. Then you see the lower part of the pictures, which is basically the symptoms. So you see what happens when a septic, sorry.
So, right, I think a sepsis with infection is now in the bloodstream, now the word sepsis comes up.
Yeah. Yeah.
Get the infection under control, all of a sudden the word sepsis stops.
No.
So how is it that people are recovering, they're not actually treating the sepsis, they're treating the infection spreading?
Okay, let's go one step back. Maybe you want to answer it, but
So like, if they're treating the infection with whatever ridiculous strong antibiotic,
yeah,
it's not actually treating sepsis.
it's not treating sepsis.
What
No,
are they doing besides giving strong antibiotics that's actually helping sepsis?
nothing. So there is no.
And how are people surviving sepsis?
There.
Why do they need?
Yeah, that's maybe you want to take that. Yeah.
The standard of care is actually supporting organ functions. So they may add drugs that stimulate the heart. They may add volume, so fluid to stimulate the kidney, for example. But that's only supporting. There are patients who will actually recover based on their own internal immune system, so they're strong enough to fight the sepsis. But in 15 to 30%, that's not sufficient. So 15 to 30% of patients who come in the ICU with sepsis, they don't have that internal strength anymore to fight the sepsis, and they will die. And actually, what Marcel just said is, well, if you have an infection, not every infection leads to sepsis. The other thing is that as soon as you have a sepsis and you have fought the infection successfully, then the sepsis reaction can still continue. So those are in the end two separate processes that are ongoing. So and that's why it is that important that you also attack the sepsis reaction itself and stop that to avoid that you have ultimate damage to your organs.
no sepsis treatment.
No. So what you basically do is you try to capture and cure the infection. That's the top part of this picture. And then the bottom part of the picture are the symptoms. As you have multiple organ damages that can happen in the case of sepsis. So you're actually trying to treat those. But that's damage control. That doesn't solve your sepsis. It's just a matter of survival. The moment you're in the intensive care with sepsis, it's a matter of survival. And you have to be a bit lucky to make it. And what exactly makes the difference between the people surviving and not, given the fact that there's no treatment, yeah, that's $1 million question, of course. But we do know that the amount of histone, the histone levels, and that's very well documented in more than 120 articles, playing a key role in survival versus death. So the patients that have high histone levels, they are actually having a much bigger chance of dying. So that's well documented. So to complete this picture then, basically where are we acting? And that's very different from any of our competitors. And we often have to actually quite explain that because people are used to treating the infection or treating the symptoms. They're not so used to having a company actually targeting the septic reaction itself. So we are basically targeting those histones. The moment they become cytotoxic, we bind them to our product. And how do we do that? We do that with a unfractionated heparin. Unfractionated heparin has been on the market since 1935. It's a very known product, has also often been used, in fact, in sepsis patients. It has been studied extensively, also retrospectively, the effect of heparin on survival, which is a slightly positive effect. However, it has one major disadvantage, and that is that it is very strong anticoagulant. That's exactly why heparin is on the market. So, but we want to have the negative, the highly negative behavior of this molecule, this polysaccharide, to basically capture those positively charged histones. So heparin is the most negatively charged molecule you can have. So what we basically do is use the heparin to catch those histones, to bind them to our molecule. And the moment they're bound, they're not anymore cytotoxic. But what have we done to make sure that we can be more effective in binding those histones? We have removed the anticoagulant part of the heparin. So basically by a separation, we take out the pentasaccharide fraction out of this molecule, and then we have a very low anticoagulant heparin, which can be given up to 20 to 25 times the amount of what has been studied in septic patients when it comes to unfractionated heparin. So that is a huge positive. And by neutralizing those histones, like I said, we break this vicious circle. That will always be on top of standard of care because you still have to make sure this infection goes away. And if there are already symptoms and organ damage, of course, you have to treat those as well. But we effectively stop the reaction that makes you die. Now, this has been studied extensively in safety studies. Of course, we can also build on a very long track record of unfractionated heparin because we don't chemically modify the molecule. We separate the pentasaccharide fraction from the rest. So we can use those data, but of course, we studied it extensively in animal studies as well. And we have done efficacy studies in animals as well. In rats, for example, we injected histones, and within 3 hours, 83% of the rats, they die. And when we give them the histones and then our product M6029, they all survive. And in mice and pigs, we induced sepsis and then gave our product where we see similar results. Whereas in mice, and that shows actually the thesis that the earlier you start, the better, we even gave it prophylactically and then induced sepsis. And you see even slightly better results in survival when you give it prophylactically. Now, that's very difficult to give sepsis product prophylactically because you don't know when somebody is going to develop a sepsis. But in an animal model, you can do that. And it is good to have those data. We also have a very good GMP production process in place. We need to scale it up, but it's working. And it's a continuous chromatography process. I don't know if you're familiar with that, but we have already tested our product in a clinical trial, a phase 1/2A combined trial in the University Hospital of Amsterdam. This was a dose escalation study, primarily focused on safety of the product, but we also look at pharmacodynamic effects. And basically, this was a 6-hour infusion that we gave to the patients. That's rather short because you know that the infection will take longer to get it under control. So you always have the risk that this whole cascade starts again. But at the time we started the study, this was the safety package we had available to go only to 6 hours. You will see later on that we will extend that for the next study. And the demographics of the patients was, it was females and males. It was people with bacterial and viral infections. And this is very important because we are looking at the histones and targeting those histones as a method of action. So it's also interesting to see, does that happen in both patients? Because we say that in our thesis, it's irrespective of the underlying trigger, how our product will work. And that's what makes it so unique. So the moment there is a septic reaction, this reaction is homogeneous. Whereas developing something in the sepsis space, often people say it's the heterogeneity of the patients that is so difficult to develop a one-fits-all solution. But we are targeting the sepsis reaction itself. And what we saw in the study is that in both viral and bacterial patients, you saw this very elevated histone levels that we were able to reduce. So that was a nice fact that we found out. We were quite sick patients, and that had to do with the fact that it was a first-in-man trial, which means that we gave a longer time for consent. You have to give consent to be in a clinical trial, but the patients were very sick, so you have to look for family members or other authorized persons to actually give the approval. So in this trial, we gave it up to 84 hours, which is very long in a septic situation, because you can die as early as 12 hours. So in the next trial, we will do that a lot shorter. But that also meant that the patients were already quite progressed in their disease stage. If you look at the most important side effect of the product or safety aspect of the product, we still look at APTT, which is basically a measure to see how the anticoagulant effect of the product is. Now, I told you that in general, we do a factor 20 of what unfractionated heparin is. A normal cutoff rate for unfractionated heparin would be around 90, and definitely in a critical condition, you can take a little bit more risk. So we took 90 as a cutoff rate. And you saw that basically only with the highest dose that we tested, and that was then also the maximum dose that we're going to use for the future, we're looking at one patient that slightly went over. But what you also see is that it goes down very fast. The half-life of the product is 1.5 hours. That means that within 1.5 hours, the half of the product is already out of your body. And that basically means that also the effect is reduced very quickly. So even though this is an important parameter to measure, we can also mitigate the risks very quickly. And it is very common for doctors to apply this, because every time they give heparin, they're already doing this. So it's not very revolutionary, let's say, approach for them, and they're very accepting of this concept. So we had no other side effects. It's very safe. We see, as we said, that the plasma concentrations are close to steady state at 6 hours. The half-life, 1.5 hours, all very predictable, also in line with what we know from heparin. And then we see also close to dose proportional pharmacokinetics. For the people who don't know what this all means, that basically means that it's very predictable what we can expect from this product. And that's very important for further development. If you then look at the histone levels, that's very interesting what we saw there. So we saw elevated histone levels, quite a lot actually, in all the patients. And then after giving our infusion, the histone levels first went up and then they were going down as the product was released from the body. Now, at first we thought, okay, how does that work exactly? But it's actually quite logical, because what happens with those histones when they start to attack the cells, they bind to cells, they are in the glycocalyx, and you don't measure them at your 0.0. At your 0.0, M69 is not yet in the body. Once we give our infusion and you see this peak, we are so powerful in attracting those histones, and we are at the moment still not able to differentiate between bound histones and unbound histones. So we are measuring the total. So the moment you add M69, you attract those histones, you measure both, and then it's released from the body. So we saw that everybody was going down in the histone levels after the infusion. We are still working on a good test to differentiate those, because that would be, of course, highly interesting to know what is the difference between bound and unbound. But that's very, very complex, and we will manage that, but we need a little bit more time for that. What was very interesting to see at the same time is that the CRP levels, and that's a very known inflammatory marker, went down extremely fast. So in the patients that received the infusions, 90% of the patients show a rapid decrease of CRP. And this is interesting because it is an inflammatory marker, but also because it is an inflammatory marker that you see in a lot of other disease areas, both in chronic inflammation as well as acute inflammation. So we also have filed additional patent protection on that as well, which opens the door for more possibilities for M629. The one patient, by the way, that didn't go down was very low. You cannot really see it in this graph, but left the ICU after 1 day. So that was not a patient that we were concerned about. We saw also a decrease in SOFA score. This is a sequential organ failure assessment score. That is basically a score to determine the fitness of your organs, which was very high with a lot of patients. We saw it go down after infusion, but we also saw it go up again with some patients after 2 days. And that has to do again with the very short time of the infusion where the inflammation is not yet fully under control, the infection is not yet fully under control. So based on these data, which are extremely good data for us to determine the further path forward and the phase 2 design that we're going to do, we're going to do a placebo-controlled trial in the next trial, phase 2 trial, with multiple dose levels. But we will extend the infusion to 120 hours. And 120 hours is about the time that you need to get the peak of the inflammation under control. So that's how we chose it together with our scientific advisory board, which is comprised of the top experts in the field of sepsis. I just told you, for the people who still remember, the complex definition of sepsis at the beginning of the story that was written by one of the advisors in our advisory board. And we have a top guy from Canada, John Marcel. We're working on a very good person from the US who's going to join us shortly as well. So that's good. And they're all behind this 120-hour infusion. We also want to start faster. As I mentioned, the earlier you start, the better. So we will start within 24 hours. So we have a more differentiation between the early stage patients that get it and the ones that don't get it, so that we see a difference there. Endpoints, we will look at histone levels, of course, because that's our method of action. We'll also look at survival, safety, organ function, organ support, 3 days in the hospital. So there's a lot of measures you can look at, and we look at different biomarkers. Of course, histones itself is a biomarker, but we look at other inflammatory biomarkers as well, like interleukins, for example. 10 minutes left? Okay, I will manage that. The trial will be executed in the US, Europe, and we're looking at Asia also. Yeah, that's basically how it will look. If you look from a market opportunity point of view, it's a huge market opportunity. It's very clear. No, there's nothing on the market. It's the biggest unmet medical need. We are focusing our business case around the 7 biggest markets. Basically, that doesn't mean we're not going to do other markets, but we don't think it's necessary for having an attractive business case. So the rest, we all treat as upside. And basically, if we reach 20% market share in those 7 biggest markets, amongst which, of course, Europe and, the 5 biggest countries in Europe and the US, we're looking at a peak sales of about 5 billion. Can it be more? Yes, of course, we will also look at other markets, but this is more than enough to have an attractive business case.
This is the market size?
No, if you look at the way this is set up, you have the TAM. This is the total possible market if you would actually give it to everybody worldwide. No, that's unrealistic that everybody would get this worldwide, but that's how you build up your market in pharmaceuticals. Then you have your sum, which is basically the share of the market, the markets you're going to target within that big pie. No, that's those 7 markets. And then we look at the sum, which is the share of the market. So in those markets that you're going to target, what is the percentage that you're actually going to reach? So the uptake in those markets. And we're looking at 20%. We call it market share, but yeah, there is no competitor. So it's market share, uptake, whatever you want to call it. But 1 out of 5 patients should get our product in those 7 markets. Now, we have a very experienced executive team. Case is by far the most experienced because he's also the oldest guy in our team. Has how much? 40 years now, drug development experience. Worked for Tibotec developing HIV vaccines, been in the infectious disease space for a long time. This company was sold to J&J. I don't like to talk too much about myself because it's not very European. I know it's very American to do that a lot, but I've been in biotech since 1998 in different commercial and investor relations roles for a variety of biotech companies that we either were sold or we bought companies also around the world. I was very much involved in the first acquisition, actually, of my first small biotech company when I started as a student, where we basically bought a company in Korea. And then the investor said, okay, that's a very nice idea. It's 10 times bigger than you, but then you also have to go there. So I went there with the CEO and the CFO. And that was after 1 year starting my career. And we had 200 Koreans, so 300, I think, all nodding at us. And we didn't speak a word of English, and we bought this company. So that's how I started. And after that, it continued. At the end, the last thing I did in biotech before I left biotech a little while was selling of Marcel to J&J for 2.4 billion. Then I decided to leave biotech a little bit. That's quite unusual for people in biotech actually to leave. I went into an industrial company where I was responsible for putting e-mobility solutions to the market in Europe. We were the first company in Europe to bring electric buses to the public transportation market in Europe. We beat Mercedes, Volvo, Scania, and all the famous brands, which was a lot of fun because we were faster, better, cleverer, much more fun to work with. And people, even the German government was fed up with the Mercedes not going fast enough with their e-mobility. So we rocked it. That was very cool. But of course, biotech is also very interesting and extremely relevant. So when I was approached by Matisse a little bit less than 2 years ago, I thought this is a great project. I mean, saving so many lives. Getting up every morning to do this and to make this work is extremely good. And with Kees, we have a great team. Kevin joined a little bit later, is also fantastic here in the US. And Peter is part of our management team. He has a lot of experience in project management and also CMC activities. Was part of Prosensa, which was sold to BioMarin, for example. On the pipeline side, I don't want to go too much into detail. I mean, we're focusing basically our product for sepsis. That's key. That's what we're looking for. That's what we're getting the funding for mainly. But we have additional patent protection on using M629 in organ transplantation, because when you take out an organ, there's also histones being released. And also when you retransplant that, it's a surgery. And in those surgeries, you have also damaged cells and more histones are released. So you can actually use that as a prevention and increase the chances of successful organ retransplantation. So there we have patent protection as well. We have also the possibility to use M629 in other histone-induced indications, as mentioned before. Histones and CRP go hand in hand in a lot of indications. You can think of Parkinson, Alzheimer. We have even in obesity, it's you see increased CRP levels. So any kind of inflammation that we could possibly target. Fair to say, we haven't really started yet to develop that part of our business, but we do have already applied for patent protection there. And then we're also working on the next generation M629 in combination with the protease, which is even more effective and faster in neutralizing histones and also cutting the histones. However, that's a completely new development. So you'll have to start from scratch also when it comes to safety studies. Now, this is the key point why we are here, and especially with you guys, we are looking for money. We have basically everything covered up to phase 2. So what we are looking for now is 40 million euro. And what can we do with that? We can complete a phase 2 trial. But at the same time, we want to upscale the production to commercial scale level because we need substantial volume of this product once it becomes available. In case of significant effect of efficacy, we are aiming for an accelerated approval because there's nothing on the market. Safety profile is very good and is highly needed. In case that doesn't work, we will do a phase 2-3 combined after the phase 2A study, for which we will then do either another round or we have an exit opportunity also after phase 2A with big pharma. We're already in discussions with quite a few pharma companies, especially the big pharma companies. They want to wait a little bit until you have phase 2 data. We are talking with mid-sized companies. They know that they have to start today, otherwise they're too late because then big pharma kicks in. So it's a bit of a game. So we're playing chess on a lot of boards at the same time with investors, with pharma. And I don't know, do you say that by the way in English? Chess on, or is that a Dutch saying? Okay, in Holland we say that. And then we aim to apply for an IND in the second quarter of next year. So we're going for the US market, we're going for the European market simultaneously. And we're going to apply for fast track approval as well. The 40 million also covers more research into 2 additional indications, but at the early stage level. So that's not the main cost. Here you see a split of the cost. So about 23% of the 40 million will go to clinical development, 22% production and upscaling. That's a big part of our cost. We have about 15% going into R&D. and the rest is G&A and there's a regulatory and quality cost to be made. So having said that, I hope that I'm still on time. Basically, what I want to give you as a message, every 2.8 seconds counts. You can really make a difference. I think if you get up in the morning and you think, I have to invest some money, where do I do that? Do I want to save lives or do I do something else? It's an excellent business case for both doing something good for society as well as a return on investment that could be extremely rewarding. Thank you very much. If there are any questions, feel free to ask. And we are here the rest of the day and also tonight. So if you would like to talk to us, no problem.
What would be the impact of your solution on hospitals' liability insurance?
What would be the?
What would be the impact of your solution on hospital systems' liability insurance?
I don't have a clue. To be honest, I haven't even thought about that because I mean, heparin has been used in hospitals forever, since 1935. So I don't expect anything different than what is usual at the moment.
Because I'm assuming hospitals are getting sued all the time.
Yeah.
For infections.
Yeah. We can't change. Let's be fair. We cannot cure the infection. We're also not avoiding the infection. We are only mitigating the reaction on the infection. So we are not going to change anything there.
Well,
However, of course, if the result is that the patient doesn't die, then there will be less of an issue.
I mean, that's got to be important to a hospital system.
we got
That gets rated for,
We had a discussion with physicians, and also last week in a conference in Philadelphia, we heard that 20 to 30% of all ankle and knee surgeries resulted in infections. The first thing you would think of is prophylactically, as soon as you perform a surgery, also apply this medication, because you will avoid the development of a sepsis.
So I can show you the scar I have from my knee surgery in June, and that was a concern.
Yeah, exactly. And you think there are so many measures in the hospital, in the surgery room, that the infection should be low, but that's not the case for whatever reason. So you can avoid that.
So I think we have a quick comment coming in here, but just before, the risk to the hospital could be on the other side, is that if this is proven to work and approved by the FDA and in hospitals around the world, they would be exposed to being sued if they don't have it, yeah, I'm sorry. Can we?
Assuming you are successful, there would still be
Well, initially, it was our plan to have a method in place, a bedside method, to measure the histones in patients, to make an upfront assessment whether our treatment would be needed or not. That appeared not to be feasible in the given time to develop it quick enough. But we're certainly working on that, because that will make your proposal stronger. So at the moment, no. In the future, it would be good also to assess the sepsis process itself during the treatment. So it will also tell you when you can stop the treatment. We don't have that relationship yet. No.
How much have you guys raised?
We have raised 8.3 million in dilutive funding and 2.3 million in non-dilutive.
So you got 2.3 on a note?
Sorry?
What was the last
Two.
price round?
The last price round was done at 28 million valuation post money. But that was before we published the clinical data.
And what's the pre on this? The pre-money?
We're looking at roughly 80 million plus minus 20%. But
What was the note? Was there like a ceiling on the note or
Nope.
20% discount or?
no, there's no note. There was no note.
There was a
No.
2.3 million.
No, that's a loan from the Dutch government.
Oh, a loan,
Yeah.
okay.
Yeah. Any other?
I might have some feedback in terms of presentation, but we'll talk about it at dinner.
This is a huge problem. I mean, Norm kind of got
Yeah.
crux of it. Actually, one of the panelists here had a pickleball accident and had a problem with his calf or something, and then he developed some sepsis infection. Following that, and had to go back into surgery 2 or 3 times more.
Very common.
So,
Yeah.
right,
Yeah.
right. So
Yeah.
it's a big deal. It's just a matter of quantifying it, and especially like, is there an insurance code for this right now? Like,
No.
how does it get paid? Right? Does the hospital pay for it? Does an insurance company pay for it? How does
So, because there's nothing comparable on the market, there is no hospital code for it. You know, right now, the hospitals and the physicians are treating the infection or treating the organs. You know, there's a road to go here, but there's a big appetite in big pharma to tackle this once we get past the phase 2. So, you know, that's going to be an issue for big pharma at that point in time, but there's certainly appetite to you know, it's $62 billion a year in ICU costs in the US alone related to sepsis. So there's a real willingness to tackle this.
That's your number.
yeah, I typically look at things in the US market primarily.
Yeah, that's why.
But that's what I said,
That's
62 in
Kevin
the
asked.
US.
Europe, something like that.
Yeah.
Last question.
Yeah, what's the duration that you've had for patients who had organ failure mitigation post sepsis? How long and what's the course for them?
The course of treatment with our product is well, what we anticipate at the moment is 5 days. It really depends on the level of damage that's done to the organs, what determines the length of the further treatment and support of that specific organ. So yeah, it's actually at the moment not possible to say how long that will be.
5 days to current, and how many patients with roughly 10 or 20 patients?
For the next trial or for what we have treated already? No, so maybe you missed that part. So in the first in-man trial, we did a 6-hour infusion. In the next one, we're going to do 120 hours. Or, of course, if the patient leaves the ICU earlier, because when they leave the ICU, we stop. In the first study, we saw that 2 patients could leave the ICU already within 3 days. So then, of course, you stop. No. So that's the maximum.
Very last
Okay.
question.